I'm a microbial ecologist. I've been at College Park now for about 2.5 years, and I'm very excited to still be learning everything there is to know about Maryland, including all of the various types of agriculture. Today, what we're going to do is take a very basic view of nitrogen mineralization. So although I garden, I have to say I lack many practical skills when it comes to things like nitrogen amendments, but I do very much understand the theory of that, and so that's what I'm going to be sharing with you for the first hour. So the outline for my talk this afternoon will be first to put nitrogen mineralization in the context of nitrogen cycling, and then to talk basically about C to N ratios and how that works and how that determines the rates of N mineralization, or if nitrogen mineralization occurs at all in systems, and then to talk about the microbes that are involved in these processes. And so we're going to start with just a view of the nitrogen cycle. I have a very simple one that I'm going to be adding to as I go through the talk this afternoon. And so we'll start off, first of all, with the plants and with two pools of nitrogen. Both of these are mineral pools, ammonia and nitrate. And of course, these are the ones that we think most about in terms of plant nutrients. And these are the pools, basically, that we talk a lot about when we think about nutrient amendments. But if we actually think about nitrogen as a whole, so I've purposely left a lot of this space blank in that soils piece, because if we really fill that in by looking overall, and so what this table shows is some of the major forms of nitrogen in soils, what we can see is that the ammonia and nitrate pool are actually really small. So that top line shows nitrogen gas, and so nitrogen is the gas that makes up the biggest proportion of our atmosphere at 70%, and even in soil environments it still makes up 20%. If we look across then at ammonia and nitrate, they make up less than 1% apiece. And this is an average over lots of systems, but very typical actually in agricultural systems is still to only be maybe at 1 or 2%. The plant nitrogen, so this would be the material that is still recognized as plants. So we're talking about the roots, for example, in this situation. They make up about 2%. And then you look at this organic nitrogen pool at the bottom, and organic nitrogen basically makes up 77%. So organic nitrogen is the largest pool. And the organic nitrogen pool then, if we come over here, basically, are any compounds in soils that basically are not any longer plant material. So this could be dead or decomposing material, or material that's from the microbial community, things that don't look like the living plant any longer, that have a carbon to nitrogen bond. And when we take a look at that, what composes that organic nitrogen pool, what we see is that there's a wide variation of compounds that have this carbon to nitrogen bond. So this pie chart is meant to kind of symbolize all of organic nitrogen. And then these different pieces basically represent the different types of compounds that when we do fractionations in the lab and when we use high-tech equipment, things that we still can recognize. So as we go around, and I'm going to click through to give you an idea of some of these compounds, at the top is nucleic acids. So this would be DNA and RNA. This is the building blocks of life. We know that every cell has DNA and RNA. And those materials, when the cells die, are released. And so for some period of time, they exist out in the soil, and we know that they make up some amount of a stable organic nitrogen pool. By far the largest, if we continue to click around, is protein. Protein is also structural and important in cells. All cells have protein. And as it turns out, proteins can vary in their ability to be broken down, and so we tend to see a lot of them stabilize and interact with mineral surfaces, for instance, so they tend to stay around for a long period of time. And then we have some pools down on this end of this, actually the next three pools that I'll talk about, both the labile, the unknown, and the acid and soluble, that really are pools that we functionally quantify in the lab. So these are made up of a lot of different things, but for instance, the labile nitrogen pool would include small compounds like urea. So these are things that are basically easy to break down. They usually only have like 1 or 2 carbons associated with them. And then we have this pie chart piece that's an unknown. And the unknown, I actually should back up one. The unknown pool here are carbon to nitrogen bonds that we just don't really know what they are. So even all of the high-tech equipment that we have, when we look at these compounds, we really can't determine or give them a particular name. And so there still is a lot of unknown in terms of what is this organic nitrogen pool. Then for the last couple of pie slices there, the acid and soluble nitrogen are these large complex compounds. This one I picked, it's not particularly common in soil, but if you know someone that suffers from high blood pressure, they might take this as a medication. So this is a compound, every one of these points where they come together, like right there, is actually a carbon bond, and those are carbon rings. Carbon rings in particular are very hard to break down, so this is something that would be around in the soils for a long period of time. And then lastly, we have amino sugars, things like chitin. Chitin is very common in fungal cell walls, and so we'll talk about a group of microbes later on, the fungi, and how they contribute to nitrogen cycling, but they also contribute, in addition to breaking down organic nitrogen, they contribute by creating compounds that are unique to their own cells, and so as their cells break down, we also have those compounds that contribute. Okay. So what I would like to do over the next couple of slides is to talk about how we go from something that is structurally complex, something like a protein that you saw in the last figure that I'm going to show up again in the next slide, how we go from protein, something that has all these carbon bonds associated with it, to the pools that we're most interested in for plant growth, things like ammonia and nitrate. And the only way that we can get there basically is through the microbial pool. And so microbes are central to this process, and they do this both internal to their cells as well as external, and it's through the processes of enzymes. And so I'm going to use the example of protein and just talk you through what some of this looks like. So again, up here we have the protein. So this is meant to basically be a folded conformation structure. So the idea here is that you have these long chains of amino acids. That's what the building blocks of protein are, and then they fold into a shape that adds some kind of structure. So you can think about amino acids as individual bricks, and the protein then being the building. So we're getting some kind of structural benefit from that. So proteins are important. They do the work of the cell. And when a cell dies or when a cell needs a new protein, they can take this structure that they've built and they can break that down. And so the first ability to break that down is to deconstruct these links. And so to break the links in between, they use a set of enzymes called proteases. And so proteases just refer to proteins, and then the ace part is the enzyme. So proteases break the protein. And what we're left with then are peptides. So peptides are these simpler structures. So you can see it there. It's basically flattened out at this point. You don't have any of those cross linkages. It's much easier to attack. And now you can actually see where the nitrogen is in that. So as you look across that compound, you see carbons, and then you see the nitrogens interspersed. So if an organism would like to use that nitrogen and use it, retask it basically into something other than protein, they can do that by creating enzymes called peptidases. Peptidases basically break those peptide chains into their single parts. So we went from basically the building to, say, a small wall of the building, now to the individual bricks. And then we can deconstruct those bricks into ammonia. And so we're back to that mineral nitrogen pool. So if you'll remember, ammonia and nitrate are actually really small pools. And so why are they so small? Well, it has a lot to do with the fact that they're used by everybody in the soil, and so they turn over very quickly. Okay. And so all of these soil enzymes that I've talked about are produced by the soil microbes, and and so if we think then about adding out some of the other arrows into the nitrogen cycle, so we went from just having ammonia and nitrate to now having both the organic nitrogen pool as well as the microbial pool, and now I've added some other arrows in just to show that basically everything is recycled. And depending on the soil system, it could be at a very fast rate. So if we go out and we measure an ammonia pool at any given time, what we find is a very, very small amount. But that's not to say that there isn't a lot of ammonia produced. It's just that these arrows often are very well balanced, and so as quickly as microbes take in or release ammonia, other organisms take and use that ammonia, either other microbes, the plants, or nitrate. Okay. So there are different organisms within soil. The most numerous group of organisms are the soil bacteria. So bacteria typically are single-celled organisms, and I have a couple different images here of them. The first is just a schematic. So this just shows a single cell, and I realize this picture kind of looks complex, but basically it's just a cell membrane. So the outer part of this is just the casing of it. The inside here is basically just the DNA. So there's no nucleus. There isn't any internal structure really. It's just the DNA. And then it's ribosomes, which are just protein-making structures inside of it. So pretty simple organisms. They are about 1/10 of our cell. So if you've ever done something where you've taken your cheek cell and looked at it under a microscope, basically these organisms are 1/10 of that size of your own cheek cell. And when you get them into the soil system, this is actually a microscope image, and the dark areas, the dark in behind is actually soil particles, and then the white material here are the bacteria. And this is amplifying or magnifying the piece of soil 10,000 times what you would be able to see with your naked eye. And so in a single teaspoon of soil, there can be as many as 1 billion different types of bacteria there, and basically ready to work on things, and they're decomposing things, and what are they really doing? They're eating and reproducing. Okay. So if I zoom in and I make a little bit simpler diagram of that same bacterial cell, and we think about what's happening in mineralization. So here's our model bacteria. And what I have next to it is a piece of that protein that I showed you in a previous slide. So this is just a little piece of peptide. You can see there that if you do the count, what you have is 5 carbon atoms and 2 nitrogen atoms. Okay. So the microbe wants to be able, let's see, we've got a message that somebody hears an echo.
Don't worry about it.
Don't worry? Okay. Okay, so basically what we have, the microbe sees this compound, and what it's going to do is mostly what those microbes want is they want the carbon. So carbon is essential to energy generation. And so the carbon really has 2 fates in this system. Okay? So what we have is they can take that carbon in from that protein. So if I go back, think about one of these carbon atoms that is going to be pulled out of that. And that carbon atom has 2 fates. It can either go to generate energy. So just like you and I, a bacteria needs to be able to generate energy. Think of this the same way that we take in calories. We're taking in carbon to get that energy every day. And so it's very similar to our own system. Basically, the bacteria can take that in. They can generate energy. And just like you and I, they're going to breathe out CO2. The alternative to that is that that carbon is going to go in and basically be used to build microbial biomass. Now you and I can do the same thing. Our New Year's resolutions typically are to not build more biomass, but we can imagine that a microorganism, when they do this, rather than put on a few extra pounds like we do, basically the bacteria can divide. And so anytime that they're taking in more carbon than they just need for energy, then they're getting ready to basically reproduce. Okay. So when they build biomass, for energy generation, the bacteria actually don't need carbon, or don't need nitrogen. So for energy generation, they only need carbon. But the same bacteria, when they're going to build biomass, they're going to build protein, they're going to build DNA, they actually need some nitrogen. And so in that case, they are going to use nitrogen. And for bacteria, the average bacteria basically needs 4 carbons to 1 nitrogen. So you're going to put 4 carbons together to 1 nitrogen. And if we go back for a second and think about this example, you can kind of see why that would be, right? So here, in this case, for this very particular compound, basically you needed 5 carbons to 2 nitrogens. And if we think about all the structures that they need to build, they actually need quite a bit more carbon than that. And so it's 4 carbons to 1 nitrogen. Okay. So if I do what we call a mass balance on this, and we actually look then at the fates. So say I have 100 units of carbon. So they could be 100 atoms. It could be 100 calories, if you want to think about it kind of in terms of energy generation like we tend to think about in human health. So basically, if we have 100 units of carbon, the energy use efficiency, so the amount that we actually need to produce energy, is about 68. So out of those carbons, 68 of those 100 carbons are going to go to producing energy, and they're going to then be given off as CO2. The remaining 32 carbons then will go to build biomass. And if you take 32 and you divide by 4, because remember, for every 4 carbon, they need 1 nitrogen. So 32 divided by 4, that means that this bacteria would need 8. So they need 8 nitrogens to go in. Well, now if I go back for a minute and I think about that same compound, if I think back to that very same compound that we were looking at a minute ago, where we had 5 nitrogens and 5 carbons and 2 nitrogens, or think about it a different way now, say, well, if we had a lot of those compounds, we would have 100 carbons and 40 nitrogen compounds. That bacterium, that single cell only needs 8 nitrogen. Okay. And so it takes in the 100, it's breaking down that compound, and it uses 8 nitrogen that it incorporates in, and then there's 32 extra nitrogen. And those extra nitrogen atoms then become ammonia. And so it's at that point, it's the excess nitrogen. So basically nitrogen that the bacterial cells don't need that becomes the pool of nitrogen that we care about. That's the pool that the plants can use. We say it's plant available, or that's the nitrogen that's been mineralized. Okay. So hopefully that's clear in terms of the bacteria and how we could make this calculation and think about it for a single bacterial cell. But of course, the soil contains lots and lots of different types of microorganisms. And so in addition to the soil bacteria, we have a whole group of soil fungi. And so fungi, unlike bacteria, most of them are multicellular. They're composed of these long filaments. And again, even though they're not as numerous as bacteria, there still is about 1 million fungi in a teaspoon of soil. And from biomass, their cells are actually bigger. So there's actually even more of them on a biomass basis. So they make up bigger structures. Okay. And then, of course, there are things that eat bacteria and that eat fungi. And so the soil animals, if we go up the food chain a little bit to think about some of the other organisms and how they might affect nitrogen, protozoa are single-celled organisms that graze on bacteria, and so you can see a picture there on the far side. And then we also have nematodes. and nematodes are multicellular little flatworms. They include grazers and predators, so they graze on bacteria, they actually graze on fungi, they actually also eat other organisms. And so we end up having what is really a very complex set of organisms to have to think about. Okay. So I want to think for a minute then about bacteria versus fungi, because although protozoa and nematodes and other soil animals are important, really the two major groups of organisms that we think about in terms of nitrogen mineralization are the bacteria or the fungi. So the fungi use carbon more efficiently, and what I mean by that is, again, I have a model cell here. So I have the cell, and again, I have that 100 grams of carbon, and in this case, 56 of that goes to energy, and now we're incorporating 48 grams of that, or 48 atoms of that actually into building biomass. So they're actually a little bit more efficient. They don't need quite as much energy to build the same amount of biomass. Okay? But there's also another difference, and that other difference is that for this 100, the amount of nitrogen that they actually need in order to build biomass now is only at 5. And so if you remember back to the bacteria, that number was 8. And so instead of now releasing 32 in the other, now the ammonia amount that's released is 35. And so I have a question there to ask for you guys to type in, and hopefully to wake you up a little bit. So will a population, if I were to put two populations of microbes into the same soil, which would mineralize more nitrogen? So which is going to produce more ammonia? A community of all bacteria or a community of all fungi? All right, so it looks like the fungi have it. Okay. So these are the kinds of experiments that microbial ecologists really do set up, where we actually do add in bacteria or fungi, and we look to actually see if this theory makes sense or not. And it actually does tend to hold, although there are some variations. And one of the big variations that we note in microbiology, let's see, so we'll go. One of the big variations that we notice in microbiology is that the relative ratio of bacteria to fungi actually changes dramatically in different kinds of environments. And so what I have here is just a range. So these boxes actually show a range of bacterial to fungal ratios that have been measured by soil microbiologists in a bunch of different environments. And so if we start down here, and I'll orient you. So this is just the fungal to bacterial ratio. So basically, the higher this is, the more fungi, right? Lower means more bacterial dominated. And then what I have here is just a bunch of different environments, some of which I've worked in. So we have Arctic tundra, places very, very high up in the permafrost, where you can see it's kind of about the middle. The boreal forest and Douglas fir forest, ponderosa pines, actually most forest systems tend to be dominated by fungi. And so that changes the nitrogen mineralization rate dramatically when we think about fungi being the dominant organisms in those systems. And then as we go on down and we go across, systems more like what we might be interested in terms of agriculture. So you see a tilled prairie there. You see agricultural soils. This is in Oregon, where I used to work. We have some prairies there. And so grassland systems tend to be bacterial dominated as well as agricultural systems. And so you can see some of that variation as you look across. So we use the information. We use two pieces of information then. We use information about the bacteria and the fungi in general and how they process and mineralize nitrogen. And then we use the information about the different rates or a different proportion or ratio of fungi to bacteria to come up with production and consumption. And so often, or if you've ever looked at the in-mineralization literature, often what you'll find is a critical C to N. And so this is the line that people often report. They give you some idea of, will you actually have mineralization, the production of ammonia, or do you end up having a mobilization? And meaning basically that all of the ammonia, as soon as it's being made, is actually consumed, and so there isn't going to be anything plant available. So in the interest of growing plants, basically we want this, right? We want ammonia, we want this. We want mineralization, we want production of ammonia that then could go to plant growth. And so this critical C to N then becomes important, because if we look below this line, then what we see is production. So carbon here is limiting. So the carbon actually slows down the microbial biomass. They can't produce enough energy and to build more biomass. And so what that means is they start pumping out ammonia. And so we then have a plant available. Above this line, that means that there's lots of carbon around. The microorganisms can continue to produce their biomass, and the microbes are going to win. In a race between the plant root or the bacteria to gain the nitrogen is going to go to the bacteria. So they are much closer to it. They're the ones that are breaking it down, and so we're relying on them to have excess if we're going to see any kind of release. Okay. So I'm going to show you numbers, and I almost didn't put this slide in because I know numbers sometimes are scary, but I wanted to give you a sense for how we actually calculate this value. Okay, so that 20, that line of the critical C to N, basically we rely on 3 pieces of information to calculate that. So the first is the community composition. So in the typical, and now I just showed you all of these ratios, so we know that there isn't necessarily a typical number, but we usually say in a typical soil, so this is usually like a grassland soil, there's about 1/3 of the population is bacteria, about 2/3 are fungi. And we make some assumptions here about how much carbon they need. So we say bacteria have a yield coefficient, so that's how much biomass that they're incorporating is about 32%, and about 44% for fungi. So remember, fungi are a bit better at producing biomass than the bacteria. They don't need as much for energy. And then C to N ratios, so how many carbons do we need to incorporate for 1 nitrogen? So remember the number was 4 for bacteria. In fungi, it actually is higher. So fungi actually have more compounds that are made of only carbon. They have things like chitin that has a lot of carbon. They have basically their cell walls require a lot of carbon. You think about the white material that we see out in the forest when you lift up the litter layer. Basically, a lot of that is just carbon. Okay. So you can then calculate this ratio by going through and saying, okay, well, if 1/3 of my population, so I'll do this, if 1/3 of my population is bacteria, and they require that 32% of all the carbon they take in goes to build biomass, and then I mix in 2/3 of the population that's fungi, 44% of it goes to make biomass, then the average for that entire population basically is 40%. So if I take 100 carbons into my system, 40 of those are going to go to build biomass. And then I make the same calculation in terms of the C to N. The average C to N, all I've done is average between the bacteria and the fungi. Okay. So I can say that for 100 grams of substrate, as I said, 60 is going to go off of CO2. 40 of that is going to go into building the biomass. And if we're going to build then 40 grams of biomass, how much nitrogen do I need? Well, we need 8 carbons for every 1 nitrogen. So I'm going to take the 40, divide that by 8, and so I need 5 grams of nitrogen. Or if you want to think about it as atoms or units, it doesn't really matter what the units are because it's 5, okay? And so then the last step is that critical C to N means that for every 100 grams of carbon, because remember that we had some of that got blown off as CO2, but for every 100 divided by 5, that's how we get that critical 20 for the C to N. And so if we're above that, we have one condition. If we're below that, we have another. Okay. So in a way of review then, if the C to N is high for the soil, so if soil C to N is high, what process is occurring? And your choices are immobilization or mineralization. And so I'll pause a minute here and let you guys think about that and type in. Okay, so it looks like most of you figured it out. In fact, I think all of you have responded and said basically immobilization. And so I would agree with that you end up having a situation where you have nitrogen immobilization. And so if we look at that, then what's happening is we have ammonia that is being produced, but as soon as it's being produced, basically it's being taken right back up by the microbes. And so I've basically marked that arrow as a red because that's the one that's actually the most important, and very little of it, in fact, none of it at all is probably going to go to ammonia or later to the nitrate pool. Okay. So the opposite then would be if the C to N of the soil is low, and that's a situation that we actually want to have if we want plant production. And in that case, then, we have sufficient ammonia that's being produced. That ammonia, some of it goes back to the microbes, but there's excess. And so that excess then can either go to, well, a few fates, but the two dominant fates are it's either going to be converted to nitrate, a process we're going to talk about here in a minute, or it's going to be available for the plants. So the trick in nutrient management, as you guys all know, is to try to get more ammonia so that we do have it available for the plants. But of course, that's a really complicated problem, or a really complicated puzzle sometimes. Because we have a lot of variation in the kinds of inputs that we can put in, and then in addition to that, then there's variation even within those inputs. So if we think about some of the things that go into soil that actually provide that organic nitrogen pool to begin with, and we think about how much carbon and how much nitrogen there is, things like cow manure actually have a lot of nitrogen, so the average is around like 15. Oh, let's see.
We'll do questions.
We'll do questions later. Oh, do questions? Okay. I'm being told we'll do the question later, so I'll come right back to that actually here in just a second. So the average C to N is for things like cow manure are 15. For corn stalks, it's 65. If we're doing wheat straw, there's very little nitrogen there, so it's 130. For soil organic matter, the C to N is usually around 10. And so this actually is a good time to answer Helena's question about when I first had that slide up where I showed that nitrogen, that organic nitrogen was 77%. That's the total percent of nitrogen in the soil. This is the C to N values that I'm showing here. So this is the amount of carbon to nitrogen. And so if we divide those numbers out, then for every 10 carbon in the soil, there's 1 nitrogen. Hopefully that got it. Okay. And so we can look at those particular inputs that I have there, but again, those are just averages. And I want to give you an idea of why that is. So think about something like cow manure, or think about some of those, plant structures. Think about a tree falling down in the forest, or you tilling in compost, that even if we have an average C to N, we still end up having all of this variation because there's still all kinds of structural components within that. And so here I just have a cross-section of those different kinds of things that we could have in some kind of mix. So think about compost mix, where we had plant material inputs, so we have things like cellulose, we have things like lignin, we have protein that's part of those plant cells, and then on top of that, in the composting process, we have things like fungi that were produced, and so we have chitin. And so even though we end up with an average, we also end up having all of these kinds of compounds that are a mix. And so again, some of these are going to break down a lot faster than others. And so the timing of that then becomes critical. Okay. And so to give you a sense for how long it takes for some of these things to actually break down, I just have some different compounds here to give you a sense for that. This is in years. So the scale here is from 0, so basically less than a year, and this goes out to 50 years, and then 100, and then 200 years, and then like 300 years for turnover. And I've just listed some of the things. So some of these are plant-derived, so the things that are inputs from the plants themselves take anywhere from about 0 to 50 years to break down. Things that are microbes, that are microbial produced, also take around that same period of time, some of them a little bit longer than others. And then we have this whole mix of other things. So remember back to that pie chart, we have all kinds of unknown compounds and things, some of which turn over very quickly in the soil, and then others that seem to take a longer period of time. And when we look then at soil organic matter in general, we have a turnover of about 50, that's that average. But now I've shown you a lot of specific compounds that are actually lower than that. And so part of what we think drives this is that there is probably some fire-derived, things like charcoal, that has some amount of nitrogen that actually gets stored, and obviously a lot of carbon that gets stored in things like charcoal that actually take quite a bit longer to break down. Okay. So I want to just tie up then talking about ammonia, and then I want to spend just a minute talking about another fate of that nitrogen, and that's nitrification, or nitrate. So when ammonia is produced, what I've talked most about is the fact that the fates are the plants are taking that up, or that the microbes are taking it up. And those tend to be the two dominant fates, but we also know and worry about in agriculture sometimes ammonia can be volatilized, so it can leave the system from the surface. Ammonia can actually also become held onto the soil particles, so it tends to stay around in soils a bit longer than something like nitrate because it can interact. The charge interacts because of that positive charge on it, and so it can get held. It can be held on exchange sites, but it also, in the soil, can be fixed eventually into clay particles. And so there it actually becomes almost unavailable to the microbial community or to the plant community to use. It could be stabilized into soil organic matter. That's what I was mentioning about things like fire-derived. And then lastly, it could go to nitrification. And so typically when we talk about in mineralization and we use the term in kind of a practitioner's realm, we're talking about both ammonia and nitrate. When I talk about it to my colleagues in microbiology, we usually are talking about ammonia because we separate this process out of making nitrate. But of course, both of those become important to the plant. And so nitrate is also produced only through a microbial process. And so nitrification is listed here. We have ammonia up here in the corner, and through a set of conversion steps, there's actually two steps in this process. The first is ammonia oxidation, and then the second is nitrite oxidation. We end up having nitrate. So as I said, this is a microbial mediated process, and in fact, there's two sets of very specific microbes that can do this. Both of them are bacteria. So the first is ammonia oxidizing bacteria. The example I have here is Nitrous ammonocherapia. And what you're looking at is this is a microscope slide where the ammonia oxidizing bacteria has been stained with a red dye. And under a fluorescent microscope, each of those little, and it's actually too hard to see the individual cells, but each of the cells fluoresce red. And so those splotches basically represent the different ammonia oxidizers. So these organisms are going from the step of ammonia to nitrite. So they're doing this first conversion. But nitrite is something that we really don't talk about very often in soils. And the reason is because we never really can measure it. So the nitrite pool very quickly gets converted from nitrite to nitrate, and that's the one we tend to talk about. And so the second organism then is a nitrite oxidizing bacteria. This is the same microscope slide that I showed a few minutes ago. And what we have here, you can see the red and the green cells are actually sitting right on top of each other. So all I've done in these microscope slides is I've just switched on a different light source. And so when the light gets switched and it fluoresces green, then I can visualize these other cells. And so we end up having both of these organisms sitting right on top of each other. And so the question from a participant are nitrifying bacteria anaerobic? As it turns out, no, they're aerobic. So they require oxygen. In fact, they are obligate, both of these are obligate aerobic organisms. Okay? So I want to give you a little bit of that information about the ammonia oxidizers. So ammonia oxidizers, they're converting nitrogen from ammonia to nitrite, and they're doing it for energy generation. So up to this point, I've only talked about getting energy from carbon. And for the vast majority of organisms in soil, that's what they do. Just like you and I, they take carbon. We eat the candy bar. They eat the carbon from soil, and that's how they get their energy. But nitrifying bacteria are unique in the fact that they actually use nitrogen to gain their energy. So they do this under aerobic conditions. They're all obligate autotrophs. And this means that they actually don't use the carbon out in soil at all. What they do is they convert carbon, they convert CO2 from the soil atmosphere into the carbon that they use. So they are not limited at all by carbon and nitrogen ratios. The only thing that limits them is whether ammonia is around and available for them to use. And so I have an example organism here, and you can see it's a simple bacteria. It's got all of these folds on the membrane, so it actually looks like a little raisin. And the reason why is because all of those little membranes are where that nitrification is happening. So they use their membranes to carry out that process. Okay? And then the second organism, here we have the nitrite oxidizers. And so similar to their partners, so these were the green cells on that microscope slide. Here nitrogen, the conversion of nitrite to nitrate is also linked to energy. So again, they're using nitrogen, not carbon. They also are aerobic. They are also usually autotrophs. So most of the time they're carrying out this process where they're fixing CO2 into carbon. They can, however, use carbon from the environment, and so they do sometimes switch if there's a lot of carbon around. This is thought to actually be very helpful to them because it actually takes a lot of energy to carry out carbon fixation. And so part of this is they're not getting as much energy as the ammonia oxidizers, and so we think this is probably because of an energy need. And then you have the picture down here of a nitrobacter. And so again, you can see all the little folding on the surface of it where they're actually carrying out nitrification. Okay. And so we think a lot about nitrate because nitrate can sometimes leave the system, and so we're very interested to understand if we have a pool of ammonia, is it going to go to the plant or is it going to go to nitrate? And so some of the things that determine that, I have just this little flow chart to kind of help you think about, well, when does nitrate actually start to accumulate? The first thing is, are nitrifiers present? So in most agricultural systems, the answer is yes. Nitrifiers are present. In the boreal forest, where I do some research in Sweden, the answer is no. We actually don't see very many nitrifying bacteria. Are there aerobic conditions? If the answer is yes, then you can proceed with it. Is there ammonia available? If ammonia is available in high amounts, because they actually like pretty high amounts of ammonia, then it can proceed as long as it's reasonable temperature. They don't like it too cold. If the pH is about neutral or a little bit higher, they don't really like acidic pH. And as long as there aren't inhibitors available, then they can do this. And so if all of those are yes, then nitrification is probable, and we'll have nitrate that then would be available for plants. Okay. So my last slide then, before I just do a summary, is I wanted to show just some data where what they've done here is this is just the incorporation rate. So this is how much nitrogen is being taken up. And it's not important to know what the scale is here, but just the relative amounts. This is in a grassland. This is in early spring versus late spring. So this was done, I think, I guess it says in February versus April. Okay, so you think about what the plants are doing in February versus what they're doing in April. And then they looked at how much of the nitrogen was going to the plants and how much of the nitrogen was going to the microbes. And the thing I wanted to point out here is basically look at all of the ammonia that the microbes take up. So they take up a lot of it in the spring. And in April, when we think about plants starting to grow, and you would think and that you could see that the plants are starting to take up more nitrogen at that point, the microbes still are the biggest pool. So they're the ones that are taking up the ammonia. And it is only because of the excess that we do have availability for the plants. Same thing in nitrate, although there wasn't as much around here, but again, the microbes actually are taking up slightly more in both of those situations. Okay. So in summary, what I hope I showed today was that the rates of mineralization are dependent on carbon inputs and on the composition of the microbial community. What we look at in particular is bacterial and fungal ratios. That ammonia has many fates, including plant uptake, but also including nitrification. And the plant available nitrogen is really just the amount of nitrogen that's left over after the microbial processes. And so I have, as a microbial ecologist, it's my great pleasure to say microbes always win. And that's everything I have today, and thank you so much for your time. So Helena had a great question, and so it's kind of interesting because it's when we talk about nitrogen application, and so you'll often hear things like, you know, he lost all the ammonia or nitrate, or that he lost all of the nitrate that or the nitrogen that he added. We are talking about those mineral forms mostly. So the net loss is usually our nitrate. I would add in there urea is something else that people sometimes are looking at in terms of what's being lost from the system, because we now know that it also sometimes can leave. But if he added carbon, would he immobilize it? And the answer is yes. So there's actually good evidence in some in restoration work, for instance, that if you add carbon into a system, that you can immobilize nitrogen. And where this has been applied is when you have invasive species that actually like to take up a lot of nitrogen, then to give the native plants an opportunity to grow, you can sometimes add things like sawdust that has a lot of carbon, not very much nitrogen, and you can immobilize in that system.